Hello everybody, welcome to this genetics playlist. We're going to talk about all types of genetics going from the basics to the general structure of chromosomes all the way to Mendelian inheritance and patterns. We're going to talk about it all in this playlist, okay? So, starting with chromosomes, this is going to be the first lecture. We're going to go in order. We're going to talk about Mendelian inheritance, non-Mendelian pharmacogenetics. We're going to talk about genetic mutations, how DNA is repaired, and then we're going to go into the molecular techniques. Not too deep, but we're going to talk about the genetics basics of it. We're talking about dysmorphology, congenital defects, diagnosis during prenatal phase, oncogenetics. We're going to talk about hemoglobinopathies such as sickle cell anemias, thalassemias. We're going to talk about inborn errors of metabolism, genetic counseling, gene therapy, and finally the approach to all these genetic disorders. So we're going to cover almost everything in this playlist. Chromosomes. This lecture is going to focus on the general structure and the normal structure of chromosomes. We're going to learn which chromosomes form the sex chromosomes and which are the autonomic chromosomes. We're going to learn how to name each specific region on these chromosomes, and it's actually due to importance because when we talk about the abnormalities, we're going to have to name exactly where the abnormality is. So let's get right into it. So here's a beautiful chromosome, and we have one chromatid here and another chromatid here, and together they're forming a homologous chromosome pair, okay? And in the center here, we have something called a centromere, and this centromere is going to split this chromosome into two parts. Everything above this centromere, we're going to call it the short arm or the P arm, so everything up here is the short arm, and everything below the centromere, we call it the long arm. So here's the long arm, and here's the long arm, okay? And we call this also the Q arm. We should both know both names, by the way. And at the ends of each chromatid, we have the telomeres, and these telomeres basically maintain the structure and integrity of these chromatids. Okay, now let's talk about the actual DNA inside of these chromosomes. So first off, we have the heterochromatin and the euchromatin. Now the main question is which one is expressed and which one is hidden. Now the expressed chromatin or the expressed genetic material is called the euchromatin, and like you can kind of guess from the name E for express, okay? And you can actually see that the color here, I specifically made it so that it's lighter color, because when you look at it under a karyogram, you'll see that the lighter part is called, the lighter part is less densely packed, like they're spread out all over the area, so it's much easier to access, which is why it's the expressed version. But when you look at the heterochromatin, you're going to see that it's much darker, and that means it's more tightly packed, hidden together, and that's why the heterochromatin is the hidden one, or the non-expressed chromatin. Now here are general definitions and terms that you should be familiar with. So pause and review these informations. Chromosomes. So we have three types of ways in which we actually classify chromosomes. We classify them based on size, classify them based on centromere position, and then classify them based on banding pattern. Size of the chromosome. So here we have our chromosomes. We split it into autosomes and sex chromosomes. Autosomes, we have 44 or 22 pairs. Sex chromosomes, we just have their X and Y, which is just one pair. Let's take it in more detail. So again, chromosomes, we split it into autosomes and sex chromosomes. Autosomes, you have 22 pairs. Sex chromosomes, you just have one pair, X and Y. Autosomes, we have, when we talk about autosomes and we want to actually characterize them, we actually number them based on the order of decreasing size. So from chromosomes 1 to chromosomes 22, we actually order them based on decreasing size. Now, if we look at this karyogram over here, we look at chromosome 1 over here, it's the largest or longest chromosome, and as we go right and downwards, we see that the size of the chromosomes are slowly decreasing. So chromosome 22 being the smallest chromosome in size. So that's the first way in which we characterize these chromosomes. Now, I want you to also zoom in on this chromosome 1 here, and we can see that again, we have our homologous pair. We have our centromere in the center, and as you can see, that's identical. It's exactly the same area, and it's exactly the same length. So that's how we know this is a homologous pair. And obviously, one chromatid came from the paternal side. This is the paternal chromatid, and one came from the maternal. So each pair has one maternal and one paternal chromatid. Now, it's important to know that these homologous pairs have specific characteristics, such as their morphology. They're exactly the same in size and structure, and also they have the exact same centromere position, and also they code for the same exact traits, so they're genetically the same. So for example, if this chromosome codes for eye color on this area right here, this chromosome is also going to code for the eye color right here. Now let's talk about the sex chromosomes. We have X and Y. Now if you have an X and Y chromosome, then you are a male, and if you have two X chromosomes, then you are a female. And there's also an important region over here, this yellow-orange region over here. And this orange region we call the pseudo-autosomal region. Now, X and Y are different, except at these regions right here, they are exactly the same and they carry the exact same information. Centromere position. Now, this is the second way in which we classify or characterize chromosomes. So as we can see here, we have our centromere right in the center, and we can see that it exactly splits this chromosome into two equal parts. So we have P is equal to the Q arm, or the short arm is equal to the long arm. And in this case, obviously the short arm kind of has a misleading name here, unfortunately. But when the short arm is equal to the long arm, in this case we call it a metacentric chromosome. And it's important to know and memorize these numbers, which is 1, 3, 16, 19, and 20. You should know which chromosomes are metacentric. Now for the submetacentric chromosomes, we have an actual short arm in this case, so you can see the P arm is actually short, and the long arm is actually long. So when the long arm is actually long, and the short arm is actually short, so long arm's P arm is greater than short arm, we call this the submetacentric chromosomes, which is chromosomes 2, 4, 6 till 12, 17, 18, and your X chromosomes. Now, finally, we have our acrocentric chromosomes, and you can see that the P arm here is super small. It's very short, and we call this actually a satellite arm, okay? And we have our long arm, it's still the same. We call this acrocentric. So these are chromosomes 13, 14, 15, 21, 22, and your Y chromosome. Now, our final way in which we characterize chromosomes is using their banding pattern. Now, the most popular technique is known as G-banding, and it's a technique that basically takes chromosomes, we treat it with trypsin, and then we digest it with trypsin, and then we stain it with Giamza stain. Meet Paul Ehrlich, a German scientist. He was very famous back in the day. He was known for discovering syphilis and also contributed to a lot of cancer therapy. So, Paul Ehrlich, imagine he came to our clinic today. He's actually alive, not dead. And he came and he presented with fatigue, and you found he has slight anemia. And when you did a blood test, you found out that, his white blood cell count is increased, but he has no fever. So, probably wondering what's going on here. You might probably think they have some sort of cancer. So you order a genetic test and order a karyogram. So how do I actually get a karyogram? What samples do I want from his body? Do I take his skin, for example? Do I take his blood? First off, we should know that during pre-birth, so for example, if it's a baby, if I want to do a genetic test on a baby, we take amniocentesis. So we take the fetus's amniotic fluid, for example, or we take a sample from the chorion or the fetal cells, and we can use this for genetic testing. Now, in our case for Paul Ehrlich over here, we will just take his blood, for example, his peripheral blood or his lymphocyte blood, or we can take his skin cells, for example. We can also take saliva, urine, biopsy cells, tumors. We can use all types of things to do a genetic test. Now, imagine we took our peripheral blood. Now, we have our blood sample. How do we actually do a genetic test? How do we look at his chromosomes? First off, we add phytohemagglutinin and we culture it in a medium. So we take all his white blood cells, we add phytohemagglutinin, and what this phytohemagglutinin is actually going to do is it's going to stimulate the T cells to actually divide. And we add it on a culture here at 37 degrees. And if you notice, 37 degrees is basic human body temperature, the normal human temperature blood. So we're basically having the perfect environment for these T cells to divide and proliferate. Next up, we wait for it to be in the metaphase. And why do we want it in the metaphase? Because it's the phase in which the chromosomes are most visible. And we add colchicine to arrest it. So we're basically saying, I want you to stay in metaphase, please stay in metaphase, and I want to be able to visualize you in metaphase. Next up, we add a hypotonic cell line, and what this is basically going to do is it's going to add it's going to basically allow the water to enter inside these blood cells and lyse it, and these chromosomes are going to spread all over the place. And we're going to finally do our G-banding, which is basically adding trypsin. We denature the protein, and then we stain it with the Giamza stain. And then we analyze our chromosome, and here's our karyotype. We just order it based on their size, right? Here we have our zoomed in karyogram of Paul Ehrlich. And as you can see here, we have something, a red arrow here pointing at chromosome 9. And if you remember before, I told you that the chromosomes pairs need to be exactly identical in two things, their structure and size. And as you can see here, chromosome 9, one pair is slightly larger than the other, and that points out or hints us that there is some kind of abnormality over here. Now also, if you looked at this arrow over here, you look at chromosome 22, and you can kind of see that one chromosome is slightly smaller than the other, pointing out that there's some kind of abnormality here. And a good doctor is going to automatically realize that a piece here probably most likely went and attached to this chromosome 9 here, and we call this a translocation, basically transported and located to a different place. Translocation, chromosomal abnormality. And this specific sequence of translocation we call it a Philadelphia chromosome, and it actually has a significant relationship to the chronic myeloid leukemia, which is basically a BCR-ABL fusion protein. And luckily for Paul Ehrlich, if he was actually in our age, we have tyrosine kinase inhibitors such as imatinib, and we can basically treat Paul Ehrlich and he might actually survive a lot longer. Now, for translocation abnormalities, we have two types, and we're going to talk about this in more detail in the next lecture, but I'm going to briefly touch on this topic. So we have two types of translocations. You have balanced translocation and a non-balanced translocation. Now, in the case of Paul Ehrlich, he had a non-balanced translocation. Imagine chromosome 18 over here, a piece of this blue part went and attached onto chromosome 19. So we basically have a piece of this chromosome, it attached here, so the genetic information here is no longer balanced, right? When this goes to the next generation, you're going to basically give a chromosome 19 with an extra piece of chromosome 18 here, and this can cause genetic abnormalities in the next generation. So this is called a non-balanced translocation. However, here we have a balanced translocation in which chromosome 18 gave its blue part to chromosome 19, and in exchange chromosome 19 gave its blue part to chromosome 18. So in this case, this chromosome abnormality is actually it's not actually visible in a karyogram. It's hard to see it because the size of this chromosome didn't actually change, right? The size is relatively the same, but in this case the size here is clearly smaller, so we can kind of tell on a karyogram, but here we can't really tell. And we call this actually a reciprocal because basically they're exchanging it reciprocally. We call it a reciprocal translocation. And in this case, we need a better technique to actually confirm our diagnosis. And this is where FISH comes in. We call it fluorescence in situ hybridization. It's a special technique where we visualize these chromosomes specially, and we can use it for example, microdeletions, rapid diagnosis of aneuploidy syndromes. Like I said before, for the culturing, we need 3 days, right, to culture for the T cells to proliferate and divide. But if we do FISH, it's almost relatively faster. And then we can also use it for supernumerary markers and rings. And in the case of the reciprocal translations, it's much more visible on FISH. So here's an example, a beautiful picture of a reciprocal translation using FISH. So you can see here it's very colorful. And you notice that we use two types of colors of probes. We use the red probe that basically lights up in red when it attaches to its target. And then we use the green probe that attaches to the target and lights up in green. So we told the red probe, I would like you to attach onto chromosome 3. So here's our first chromosome 3. The red probe attached to it, found it, all good. But when it went to attach to the other pair, right, we have two pairs of chromosomes, chromosome 3. When attached to the other pair, chromosome 3, we found that it was kind of split, right? We have a small piece here and a slightly smaller chromosome 3 piece here. And we then decided that, okay, let's also use another probe, a green probe, to attach to chromosome 20, which is the green one over here. And we found a piece of chromosome 20 on chromosome 3, and the probe also found a piece, a light lit up green on chromosome 3. And we can immediately tell here that a piece of this chromosome went and attached onto chromosome 3, and a piece of chromosome 20 went and attached onto chromosome 3 here. So it's immediately obvious that there's a reciprocal translation going on here, and this was using FISH, right? Now in the second example here, we actually wanted to diagnose a microdeletion syndrome. So what did we do? Again, we had two probes. We had a green probe and we had an orange probe. Now we used the green probe as a control probe to actually identify that these two chromosomes are actually the same chromosome. So the green probe attached to this chromosome, the green probe then attached to this chromosome, which is chromosome 7. Now we confirm that these two chromosomes are actually pairs. Then we added our orange probe and we're looking for the microdeletion. So if there's a microdeletion in, like, for example, one chromosome, the orange probe would light up if it found that gene. But if it didn't find the gene, then that means that certain gene is deleted. So you can see here, we found the microdeletion here. So there's no actual deletion here, right? But when it went to attach to the gene over here on chromosome 7 on the other pair, it did not find that part to attach to, which means that this part might have been deleted. Actually, it has been deleted, confirmed, which means that it's associated with, for example, Williams syndrome, which is deletion between 7q11.23. So that's just another way in which we can use FISH to diagnose microdeletion syndromes. Now, the final part of this lecture, we're going to talk about how to actually name certain regions on a chromosome. So let's first start with the actual chromosome itself. So this chromosome, this entire chromosome here, we know is chromosome 7. So we just place it here, 7. Okay. Next up, let's zoom in a bit more. Let's choose which arm. Let's identify or clarify which arm we're actually looking at here. Are we looking at the short arm, or are we looking at the Q, or the long arm? Now, in this case, the red circle here is pointing at this region over here, which is actually in the long arm. So we're gonna throw a Q right here. So that's 7Q. Next up, let's zoom in a bit more. Let's actually designate a region. So next up, let's take a designated region. How do we actually name these regions? We start at the center here where the centromere is, and we count region 1, region 2. If there was longer, it would be region 3. And here again, from the centromere, we count up region 1, region 2. So here it's actually in region 2, so we plop a 2 here. So we have 7, chromosome 7, long arm, which is the Q, and then region is number 2, right? And then finally, we're going to actually identify the band. So the bands are basically at the top of the region, 1, 2, 3, 4, depending on which bands are there. And in this case over here, we also have 1, 2, 3, 4. So here it's band 3. So this is how we name it. Chromosome 7, that's the whole chromosome. Long arm, so 7Q. Region 2, so in this region, band 3. This is not 23, this is region 2, band 3. So when we talk about a cytogenetic karyotype, for example, when there's actually a abnormality, we actually will use the number of chromosome, then we identify the sex, so it's XY, and then we add the abnormality. So for example, if it's a translocation, we add T. If it's a Robertsonian translocation, we add a Rob. Each type of abnormality has its certain keyword to it. So deletion would have DEL, del, for deletion. So here we have chromosome, the total number of chromosomes, so 46, and then we add the sex, then we add the abnormality, and then we have the chromosome abnormality, or the actual region on the chromosome, which is what we learned over here. We have chromosome 5, the short arm, so P, and then region 1, band 5. So region 1, band 5. So I hope that clears up the naming. And in the next lecture, hopefully we will talk about the structural abnormalities that actually occur in each karyogram. We're going to talk about all types of examples.